My name is AJ Bushy. I'm in the communications department here at BTI, and we're going to start some poster presentations here at lunchtime. And we will start with Elise. Hello.
Hello, how are you?
Pretty good. How are you?
Good, thank you.
So this is Elise Boisvert. Did I pronounce your name correctly?
Yeah, thank you.
Excellent. Could you please tell us whose lab you're in and tell us a little bit about your project?
Absolutely.
And speak into this mic here.
Certainly. I'm in Margaret Frank's lab, and my project was to explore the role of root architecture and suberin content in salinity tolerance in tomato rootstocks. So when I started in the lab, I was paired with a grad student, Brandon Williams, and he was working on a project to develop a rootstock that's ideal for local cultural practices and growing conditions. And this makes sense because current rootstocks that are commercially available were developed for use mostly in hydroponic systems, whereas local growers overwhelmingly grow under high tunnels. So in order to do this, we developed 36 successions by crossing M82, which is a common research tomato, with Solanum habercites, which is a wild tomato relative. And in doing this, we get traits from both parents as well as the benefit of hybrid vigor. You may be wondering what any of this has to do with salt. So soon after I started that research, I was encouraged to apply for an undergraduate research grant. And so I started reading. And while perusing the literature, I found that the current method that we use for predicting salinity tolerance in a rootstock, which is ionomics profiling, may not tell the whole story. So ionomics profiling is where we take a sample of the leaf and we find the level of different ions within that sample, and that includes salts or sodium chloride. And in order to tell you why that may not be ideal, I need to explain the concept of excluders and includers. So excluders are saline tolerant plants that use their own sugars in their leaves to prevent the osmotic uptake of salts from the soil. And this actually can be detrimental to growth because the sugars are then tied up in that process rather than being used for growth. And then we have includers, and includers actually uptake some salt from the soil and put that into the leaf in order to prevent further osmotic uptake. And they may seem to have very high levels of salt in ionomics profiling and still be remarkably salt tolerant. So to put it another way, some salt tolerant plants have tons of salt in their leaves and some have very little. And so I wanted to come up with a different method to predict the levels of the salinity tolerance in our lines. And so I thought about a few of the other ways that plants mechanisms for salinity tolerance. And one of the ways that plants prevent salts from coming in is at the point of the Casparian strip. So having a suberin-rich Casparian strip can be really, really helpful. And another way is simply by sequestering salts in the roots. And so my hypothesis is that rootstocks that have vigorous root architecture and high levels of suberin are going to be grow better under saline conditions. So in order to test this, one of the things we did was develop rhizotrons, and we use these for imaging the root systems. This is just a mock-up. I planted it yesterday, so we cannot yet see the lovely root system in the back. But we use these and then put our roots through Rhizovision Explorer, which is the root phenotyping software to help us analyze the roots. And then the next step is to perform confocal imaging for suberin analysis. So we will take cross sections of our roots and stain them and use the confocal microscope to find the level of suberin. And finally, we'll actually plant our plants in salt conditions. So we'll plant 5 replicates in salt and 5 in under normal conditions. And we'll measure the differences in growth using a few different measures of success, including the overall root architecture, the dry weight of root and scion, and the level of salt in their leaves.
That's really fascinating. And this is really cool. Is this something you came up with yourself?
I did, yeah. So we used Petri dishes, and I actually used a table saw to cut off the tops and to cut drainage holes in the back.
That's awesome. You gotta love the real science in action. That's how the sausage is made, they say, right?
That is what they say.
Can I ask you, what was your favorite part about the summer here?
That's a great question. I really enjoyed getting the opportunity to work really in depth on research without other distractions. Not having to think about classes and being able to like really go in and focus every day on research was a phenomenal experience.
Yeah, that's great. Do you think you'll stick with science as you continue going forward in your career?
I do. I'm working on applying to grad school as we speak.
Well, that's great. Congratulations, Elise. This is a fantastic presentation.
Thanks. I appreciate
All right.
that.
Thank you so much. And we'll move on to our next poster presenter. Hello, this is Lucas Jender. How about I move this way? Okay. And could you tell us where you're from, Lucas, and whose lab you're in, and what research project was this year?
Hi, so my name is Lucas Jender. I was in the FAE lab this summer, and my research project was the de novo genome assembly of Solanum neurickii and the identification of structural variants between neurickii and the cultivated tomato. So the tomato is one of the most important fruit crops in the world, and in the past, wild species have been used to introduce desirable traits like disease resistance or larger fruit weight to the cultivated tomato. And one such wild species is neurickii, which we looked at in the study. In the past, there was a study that created a backcross inbred population between neurickii and Lycopersicum, and identified locations based on genes where there could be desirable traits, quantitative trait loci, so QTLs, and then, but because there's no full-scale assembly of the Solanum neurickii genome, we don't know the exact structural variations that account for these traits. So the first part of our study was to assemble the Solanum neurickii genome. This was done using PacBio Hi-Fi reads, which compared to previous technologies have a similar 99% accuracy, but a much longer read length. In total, we identified 225,000 structural variants between the wild tomato and the cultivated tomato, Solanum lycopersicum. And we focused on one region that controls phenylalanine, the phenylalanine ammonia lyase gene. In total, we found 2 insertions and 3 deletions in this gene, and they were all within the regulatory region of the gene, meaning they could affect the gene expression, which would in turn affect phenylalanine levels in the plant, which could then affect the fruit quality in the plant. And so further studies would have to be done to identify exactly what these structural variants are causing. And then in addition to that, the assembly and the structural variants we identified between the 2 genomes could serve as a valuable resource for future genetic and molecular studies.
That's really cool. And could you tell us what was your favorite part of the internship this summer?
I think my favorite part was working in a more formal research environment and learning exactly how the research project works, and then working with my mentor to go through each step.
Yeah. And this is more bioinformatics, correct?
Yes.
Yeah. So do you think you will continue on in that in some form in the future?
I think that I'll definitely consider pursuing a career in either computer science or biology. I'm not quite sure yet.
All right. Well, I know you have a good future, so good luck to you.
Thanks.
All right, on to the next poster. This is Sophie. Hi, Sophie. And Sophie Murtha. Did I get your last name right?
Yep.
Could you please tell us a little bit about where you're from and what your project was this summer?
Okay. I was working in the, I'm from Ithaca, New York. I was working in the Giovannoni lab, and my project this summer was I was doing metabolite extractions for nutrient composition and gene expression analysis. So we were looking at tomatoes because tomatoes have a really wide range of phenotypic diversity, especially in terms of color. So we looked at 19 different tomato accessions, some wild species, some heirloom and modern tomato species. And we wanted to see their carotenoid profiles because, well, carotenoids and chlorophylls are the major pigments in tomatoes responsible for like their coloring. So we looked at carotenoid profiles in tomatoes to see how they were in tomatoes of similar and different coloring. So as expected, we found that lycopene was the major pigment in red tomatoes, and beta carotene and lutein were the major pigments in yellow and orange fruits. But we saw one tomato, like one of the cheese mini tomatoes, which is yellow, had a really high level of beta carotene, and it had some zeta carotene, which is interesting because if you compare it to one of the cerasiforme fruits here, they're both pretty similar in terms of their phenotype. They're like their yellow color, but the one cheese mini fruit had a lot higher accumulations of beta carotene. We also looked at chlorophyll levels in mature green fruits. And in heirloom tomatoes, we found that chlorophyll levels were higher if there was a green shoulder, which is like the green on the top of the tomato here. We also looked at chlorophyll and carotenoid levels over the course of the ripening of fruit number 142. And as we expected, chlorophyll levels tend to decrease as a fruit ripens, and carotenoid levels increase. And throughout this process, we're also looking at the extraction efficiency of THF and ethyl acetate, because carotenoids have varying like levels of, or mostly nonpolar, but there's varying levels of how nonpolar they are. So we wanted to see if using extraction solvents of different polarities would affect that. And we generally found that ethyl acetate was a better extraction solvent and more efficient at extracting carotenoids. We did some gene expression analyses of carotenoid biosynthesis genes. So we looked at how, like, which genes were expressed in 12 different tomato accessions. So what was interesting was that in the cheese mini fruit, most of the yellow fruits that have lower levels of most carotenoids, but still have some beta carotene and lutein, we found that the lower levels of carotenoids aren't due to low production of carotenoids, but rather more downstream degradation. So here, because CRT iso is normally expressed, and CRT iso is responsible for producing lycopene, but then LCYB and CRT or epsilon, they're genes that are downstream that are responsible for converting like upstream products into like more yellowish and orangey pigments. So that was it. Yes.
That's a lot of cool stuff you figured out this summer. Congratulations. What was one of your favorite parts of being here for the summer at BTI?
I really like doing all of the carotenoid extractions and working with like pipettes and doing running gels, and that was really interesting.
That's great. So the nitty gritty, the fun stuff.
All of the wet lab stuff and like technology, that was really cool. Yeah.
That's great. So how do you think this work may influence how you what you choose for a career in your future?
I think it definitely put me on the path to being interested in like research and just how in like college I'd like to find more opportunities to pursue research, and yeah.
That's great. Well, congratulations. This is a fantastic presentation. Okay, we'll move on to the next poster. Number 4 is behind me. Here's Tim. And Tim Costfield. Did I pronounce that right? I will get on this side, I think, so you
Sounds
can.
good.
So that look good.
Hold this. I feel bad that you have to go presentation to presentation.
If you feel comfortable doing it, that's fine.
If your arm gets tired, just let me know and we can switch.
So could you just tell us your name, whose lab you're in, and about your research?
Sure. I'm Tim Costfield. My wonderful mentor is Anna Hermans, and I'm in the Dr. Giovannoni lab. We created VIPER, which is an acronym for Variant Identification Pipeline with Expression Realization. That's kind of a stuffy way of saying that we built a tool that finds structural variants and annotates them for function. A structural variant, the definition is a little contested, but it's generally accepted as any genetic permutation, like an insertion, a deletion, a duplication, greater than 50 base pairs long. What makes structural variants unique is that A single structural variant is enough to have a significant impact on the phenotype or the appearance of a plant, whereas the more commonly studied single nucleotide polymorphisms would have to manifest thousands of times before possibly seeing a result. This means that Viper, which accepts RNA data and outputs a library of significant and labeled structural variants, has the unique ability to identify genetic variants underlying desirable phenotypic traits. We tested the efficacy of Viper on 36 tomato accessions with 3 biological replicants per accession for a total of 108 samples. RNA data was taken from the leaves of these accessions, put through a pipeline developed in lab to clean the RNA data and output normalized RPKM data, which is what Viper accepts as input. Associated with these RPKM values are 35,000 total genes. These genes were cross-referenced with the publicly available structural variant data set, which allows us to pair genes and variants through mutual positioning, whereas the structural variant and the gene can be paired together if they overlap in chromosomal position. Of the 35,000 total genes we were working with, around 5,000 successfully overlapped with the structural variant, and of those 5,000, 3,500 manifested in enough tomato accessions that they were considered viable for statistic analysis. Statistic analysis is done on a per SV gene pair basis across an accession split, a divide between accessions that do not have the structural variant and those that do. By measuring the differential expression of the genes, those without and those with, using a Wilcoxon rank sum test, we can output a p-value that's used to rank the SV gene pair amongst its peers. If that p-value is normalized below 0.05, then the SV gene pair would be considered significant and be annotated to a library for further identification. Of the 3,500 SV gene pairs we analyzed, 271 were considered significant or high impact and moved on to identification. During identification, 103 genes were identified as relating to leaf development, with the highest ranking of these 143 base pair insertion we labeled gun gear, expressing very powerfully in terms of phenotypic influence over leaflet shape. Using parameters developed by the Sinha lab at UC Davis, we were able to see that any leaflet that possessed gun gear expressed significantly differently than those without for 5 different principal components, which is more visible in the accessions here to the naked eye, where these leaves do not possess gun gear and they're quite circular, but those that do have a less uniform aspect ratio, they have a reduced tip base differential and an increased serration and lobing density. They look a bit more like a spear. Now, while leaf morphology is an excellent proof of concept for Viper, our focus moving forward would be to use its ability, now affirmed by gun gear, to create libraries of candidate SV gene pairs to target agricultural factors. Those can include carotenoid accumulation in tomatoes for increased nutrition, hormonal control, hormonal metabolism, fruit flavor, fruit shape like morphology, or fruit ripening. And that's all I got.
All right.
Thanks
That's really cool. So quick question, is the Viper, is it like a software, computer software?
It is computer software.
Gotcha. Okay, well that's really fascinating. You did this in Jim's lab.
I did.
Yeah, that's great. Could you tell us what your favorite thing about the summer here at BTI was?
As odd about, it was mostly spent doing computers, but the highlight was going in the fields and working with tomatoes. My lab is full of really wonderful social people. So going out in the nice 70-degree Ithaca summer with some friends and just a couple hours and planting some plants was a lot of fun.
Yeah, that's great. That's great to hear. And do you think this will impact what you do in your career in the future?
Oh, definitely. I think so. First time interacting with plants, and I had a lot of fun, and I hope to stick around.
Do you normally do computer science or?
I do computer science with a focus on immunology. So this was a bit of a vacation.
Well, it's great work and a very good presentation. Thank you so much. All right, we have one more before our next break. Pardon. And here we are with Albert. Hello, Albert.
Hi.
Albert's son.
This is for.
And could you please tell us a little bit about yourself, where you're from, and whose lab you're in, and what your research project was this summer?
Yeah, my name is Albert Sun. I'm from Ithaca, New York, and my mentor is Dr. Lucas Mueller. And this summer, I worked on the breed-based website. So basically, in recent years, the focus of breeding has shifted from kind of looking at desirable traits and just crossing them with others to more digital analysis of genomes and phenotypes. And so basically, what BreedBase does is it wraps all of the digital analysis tools into one single system. And it's really easy to access because you can just access it through a web browser. You can visit it at like breedbase.org. And so I basically worked on writing new tools for BreedBase this summer. The two tools I wrote are the radar chart tool and the progress tool. So basically the radar chart tool, it lets users select certain traits and accessions, and then it'll then compare these traits and accessions, or it'll compare these traits with other accessions. So for example, if you select like two data sets here, you can hit render and then it'll compare it with another data set. And then, yeah, so basically it just allows breeders to compare different accessions of a certain plant.
So each point is a different accession?
No, so
Or each point is a different trait?
Each point is, yeah, a different trait. So I'm comparing two different accessions of banana here. And I'm just comparing the average finger length in different cycles of life. And then the second tool I worked on is the breeding progress tool. So basically the progress tool allows breeders to select certain traits for a certain breeding program, and then it displays the progression of that trait over the time period like of the breeding program. So if I go here, I guess, so this is like the finger circumference of a plant. This is Musa, I think. And then it just displays the progression of that plant over the period 2012 and 2018. And it just shows that the finger circumference has increased between 2012 and 2018. And so both the radar chart and the progress tool will allow breeders to easier analyze phenotypic data and also determine how efficient their breeding programs are going. Yeah.
That's great. And what was your favorite part of the summer program here?
Yeah, I really enjoyed learning new tools, and I also learned a lot of new techniques, and yeah, I learned a lot of like new skills like SQL and libraries like Chart.js and Vega. Yeah.
And it sounds like you've made some real tools that will be used all over the world to help improve breeding programs. That must feel pretty good, right?
Yeah, I guess. Yeah.
And do you think this will, summer program will influence what you do in your career?
Yeah, I think it's gotten me more interested in like bioinformatics in general and like biology in general. So yeah, I might consider like going down that road. Yeah.
Were you mostly interested in computer science when you came here?
Kind of. I was also like just curious about like what it's like to work in bioinformatics, and I thought it would be fun to do this program.
All right, was it fun?
Yeah, it's pretty fun.
That's great. Well, thank you so much, Albert. You did a great job. How you doing?
Pretty good.
All right. Yeah, pretty good. Okay, we're back here at BTI at the George and Helen Kohut Symposium, where we're celebrating our undergraduate and high school research interns. And here we are with Tony Mao. And could you please tell us where you're from and a little bit about your research program this summer?
Yeah, I'm from Ithaca. I go to Ithaca High School. And this summer, I have been starting a project exploring the genetics involved with domestication in the Solanaceae family. And basically, the Solanaceae family is a family of over 2,500 species. and it includes important domesticated plants such as tomatoes, peppers, potatoes, eggplants, etc. And basically for my project, I'm utilizing bioinformatic tools to explore positive selection and selective sweeps within domesticated and wild groups of these plants. And so like tomato would be one group, pepper would be another group, and so on. And basically for my project, I've been focusing on tomato, wild and domesticated group because it's like the most known group and it can be used as like a control to test these methods for eventually other species. And basically how I did it, how like I explored this was I found genomic data off of databases and ran bioinformatic tools such as OrthoFinder, which constructed like sequence alignments of genes within the sets of within like the tomato group. And these alignments were edited to then run through a Perl script, which then led to an CodeML output. And CodeML is basically a part of PAML, which is a PopGen analysis tool. And CodeML produces omega values and for each alignment, and then those alignments with omega values greater than 1 indicate positive selection. And then those alignments can be found on this table. And the other side of the project, which is like selective sweeps, I used an existing VCF file, edited it to remove all wild species except for one, and then ran RACE, which is a different program, on that VCF file, which basically identifies regions that are indicated with selective sweeps. And I have a tree with like the relations of these species to each other. So tomato, potato, eggplant, and basically I've been focusing on this group of domesticated and wild tomato species. And so as a result of the positive selection and selective sweep, we were able to identify 3 gene groups with functions that could be connected to domestication. And they're labeled on these graphs, the selective sweep graphs, to basically connect positive selection to selective sweep within tomatoes. And the functions of those 3, MADS box, which is been shown to affect fruit size, BHLH95, which has been shown to affect germination and flowering, and then defensin-like proteins, which are related to pathogen defense. And then there's another function, WUSHL, which has been previously known to be related to domestication, and that one's also labeled as basically a reference. And then basically the next step, the ultimate goal would be to expand these methods to other groups of species like peppers and eggplants, to basically compare that to tomato and identify genes connected to domestication, which would help improve breeding and eventually like crop growing in general.
That's great to be able to help out crop improvement. What was your favorite part of working here this summer?
I think my favorite part is really just getting to know like what bioinformatics is like in a professional setting, and like all of this was pretty new to me, like Linux and all these tools. And then I also really enjoy just working with my mentor and like other people in my lab, because collaboration is nice.
So will this influence what you do, what you think you'll study when you go to college?
I've definitely been interested in like computer science. I think this experience will really like make me think about connecting computer science to other fields such as biology.
Well, that's fantastic. That was a great job.
Thank you.
All right, thank you so much, Tony. We'll move on to our next person.
Then there's, sorry.
Hello.
Then there's at the beginning. So
How's it going today?
good. How are you doing?
I'm doing pretty well. Okay, I'm here with Anna Duke Cadenas. Did I pronounce that correctly?
Yep.
Great. And could you please tell us where you're from and whose lab you're in and what your project was this summer?
So I'm from Ithaca, and I was in the ski rich lab. So my project was looking at characterizing stress granule components under both salt and heat stress. So stress granules are non-membranous organelles that form when a plant is under stress. Importantly for my research, cyclin-dependent kinase A, which is a growth regulator, is sequestered into stress granules, which means it's not available to the rest of the plant and could suggest a connection between stress granule formation and growth regulation when a plant is under stress. There was a sort of second part to my project, which focused on an RNA binding protein known as RBP47B. And because that has been reported as a stress granule marker, the Skirich lab was using it under a promoter called 35S to characterize the link between stress granule formation and growth regulation under both salt and heat stress. But 35S does induce a really high expression of the gene, so that could affect results. So for the RBP47, I was supposed to look at two different lines of Arabidopsis that had RBP47 under 35S, the promoter 35S, and under a native promoter, and compare stress granule formation under both lines, under both promoters, to see if there was a difference. Unfortunately, I was supposed to do that and visualize the stress granules looking under a confocal microscope. And there was no signal. So this is actually the black square is just the image of there was no signal that came from the root. So unfortunately, because of time constraints, I wasn't able to pursue that. But we were able to go through more with the CDKA because something similar happened. The original line I was supposed to be using had a really fuzzy signal, so I wasn't actually able to use it for my project. But we used a mutant line, which has CDKA1, and in that we tested to see if CDKA was sequestered only into stress granules only under heat stress, or if it was also under other stresses, and we specifically tested for salt stress. So the first two images are heat stress. The first one is for half an hour, and the second one is for an hour. And you can see those dots of more concentrated yellow. Those are where the stress granules are, and the CDKA is in them. This is also heat stress, so you can see the stress granules there as well. That is what we expected. So far, going according to plan. The second two images are under salt stress. So this is salt stress for half an hour and salt stress for an hour. And if you look here, it is more smooth than what you see here. This is also more smooth than what you see here. And when you compare it to the control, which underwent no stress, it looks pretty similar. And that's because the CDKA didn't actually go into the stress granules under salt stress, unlike what we expected. So we do know that stress granules would have formed under those conditions for salt stress, which means that CDK simply just didn't go into the stress granules. So that could indicate that CDK being sequestered into stress granules is not conserved across different stresses. It's specific to heat stress. And so in future directions, we could check to see what mechanism is used for growth regulation under salt stress. And we could also see what happened to the signal, the fuzzy signal that I got before, and see what caused that, and if there's a way that we could fix it. Same goes for RBP47, unfortunately. We would also have to check to see what happened with the signal there, because other plants under that line had expressed a signal. So this was sort of an anomaly, and we'd have to see what was up with that and what had changed.
So those be your next steps if you're going to continue the project?
Those would be the next steps, yeah.
So what was your favorite part of being here at BTI this summer?
I think like meeting all the people and seeing all the different directions that research could go, because like, I mean, you've already seen other posters, like those are completely different projects to my own. I wouldn't necessarily be able to understand them unless someone else explained them to me. So just seeing how many like different approaches to similar problems can be found in the same building done by different people. I thought that was really cool.
Yeah, that is a great thing about being here at BTI. Do you think this research project will influence what you do in the future?
Oh yeah. So I'd always been interested in science. I wasn't sure if I was interested in research, and this was a really great opportunity for me to find out if I was. And I think this was especially good because not everything went according to plan, which really like shows, and it doesn't, like it rarely does, and like that's like kind of opened my eyes to like how complicated research can be. And I'm glad because now I can have a more full understanding, and it can like help me decide if this is what I want to do in the future, because I know I'm interested in it now and I wasn't sure before.
Well, that's great. Well, congratulations. I hope you had a good summer.
Yeah, I did.
And it was a great presentation. Thank you so much. Okay, we'll move on. Excuse me. Okay.
Am I next?
Are you Sadvi?
Yes, I am.
Yes, you are next.
Okay, great.
Sorry, George. Could you let our camera person to? Thank you. Okay. All right, we are now here with Sadvi Mohan Kumar. Did I pronounce that correctly?
Yes, that's right, you got it.
Could you please tell us where you're from and what you did this summer?
Yeah, so I'm from the College of New Jersey. I'm from New Jersey. And this summer, over the last 10 weeks, I've been studying the role of the enzyme IAA amidosynthase in mediating plant defense responses through conjugation of plant hormones and amino acids. So speaking in a broad sense, corn is a really important crop because it's a staple food crop and also because it's the largest economic agricultural driver of the United States. So learning more about the defense pathways of corn will really allow us to develop natural deterrents to pests such as the fall armyworm caterpillar that currently wreak havoc on our crops. And this is really important to sustain both agricultural efforts and the human population's demand for more food. So a little bit of background behind corn defense pathways is jasmonic acid is a known plant hormone that is conjugated with isoleucine and becomes an active defense regulator. And indole-3 acetic acid, on the other hand, is a known growth plant hormone. So it actually suppresses growth when conjugated with isoleucine. So what I hypothesized was this enzyme IAA amidosynthase can simultaneously activate defense and deactivate growth at the same time. And the reason I was led to hypothesize this is because these two plant hormones share similarities in their structure. So they both have a carboxylic acid side chain. And because of the fundamental growth defense trade-off, defense pathways require a lot of energy, and plants can conserve energy by reducing growth. So in order to test this hypothesis, I first knocked down the IAA amidosynthase gene using VIGS. So VIGS is virus-induced gene silencing, and I did this in two different methods. The first method was biolistic delivery. So I transformed this gene into a gene of interest into the VIGS sugarcane mosaic virus vector and transformed it into E. coli. And from there, I was able to bombard the plants using a gene gun to infect them. And then the other method was through injection, where I used agrobacteria instead of E. coli, and agro-inoculated the seedlings using a syringe. So this virus silencing took 2 to 3 weeks to occur, and at which point the plants that were correctly infected had this mosaic infection phenotype on the leaves. So once I was able to collect the correctly infected plants, I measured gene expression through qPCR. The qPCR results showed a general trend of reduced gene expression. So you can see there's a 50% reduction in gene expression in the target plants compared to the control plants. So there was a lot of variability though because of a small sample size. However, the caterpillar bioassay results were much more promising because the caterpillars recovered from the target plants were over twice as heavy as the caterpillars recovered from the control plants. So this indicates that the caterpillars perform significantly better on the target plants because there is a downregulation of defense pathways. So in conclusion, these results indicate that the IAA amidosynthase gene is likely involved in defense pathways. However, further research is necessary to draw specific conclusions on its exact role. So in the future, what I'm planning to do is increase this sample size substantially by I've collected the plant tissues from the correctly infected plants and stored them in liquid nitrogen. So now I can ship that back home to New Jersey with me and inoculate many more plants so I can more accurately infect a lot more. And then I can do all sorts of tests on it. Yeah, so thank you, yeah.
That's fascinating. You will get to continue this on back at your home university.
Yeah, that's the plan. So actually, VIGS is a technique that we don't use currently at my institution at home. So I'm excited to be able to teach this to the other students in the lab and kind of work on expanding this process in other universities.
Oh, that's excellent to hear.
Yeah.
So what was your favorite thing you did here at BTI this summer?
So I've learned a lot of new lab techniques. I guess being I'm a rising junior in college, so most of my college experience has been virtual due to the pandemic.
Right.
So this is kind of the first time I've actually been in the lab and working with wet lab and experiences and working with actual caterpillars and bagging plants has been a completely new and amazing experience.
Yeah, that's one of the things with science is you really need that hands-on experience, huh?
Yeah, like I've read a lot of theory, but nothing compares to actually doing it in person.
Well, I'm so glad you were here to do it with us.
Yeah, thank you so much for having me.
Right. And congratulations. This was an excellent project and some really cool results.
Thank you so much.
All right.
Yeah.
All right.
Thank you.
And Miguel Barrera? We're good to go? We're not good to go yet? 1 second. Maybe I'll step over here. All right. So Miguel Barrera, welcome here this great symposium. Could you please tell us where you're from and what your research project was this summer?
Sure. I'm from Cornell University and I worked in the Jender lab. And I was focusing on an aphid effector protein in Nicotiana bentamiana, which if you don't know, it's a cousin to tobacco. So it's smaller, but it's easier to manage in a laboratory setting. So I can't tell you the amount of times I've come back smelling like nicotine. Yeah, but pretty much my research has been focusing on 2 transcription factors in Nicotiana. And these transcription factors are hypothesized to bind to an effector protein. So this protein is found in aphids and it's used to infiltrate the plant pretty much. So they secrete it in the saliva and then they inject their mouthpiece into the plant to get the nutrients in the sap. And if you look at image 1, this is what my mentor actually discovered, that the SP15 effector protein localizes with 2 partners in the nucleus, which we hypothesize are the 2 transcription factors I'm studying. And SP15 manipulates, or we speculate that it manipulates the gene expression of the plant. So how the plant increases or decreases its defense, right? So we want to understand the pathway, the bigger idea of what's going on here. So in order to do this, we have to knock down these 2 transcription factors, and we're doing this by using a gene editing system. So bypasses the need for tissue culturing, which takes a whole year. This could be done in a few months. So my method here shows what I've been working on over the spring. I actually started this project back in February when I started in the lab, and I've been continuing it over the summer. So in the first part, it shows that I designed a guide RNA, which pretty much tells CRISPR-Cas9, an enzyme in the plant, where to mutate, where to cut, right? And I've fused it to an FT RNA, which I'll describe in a moment what that's for. But once I designed the sequence for each transcription factor, I cloned it into a TRV vector, which is known as tobacco ratovirus. So it's a virus that scientists use to infect plants and inject whatever DNA they want to express, right? And the problem with tobacco rattle virus, not tobacco rattle virus, rather virus-induced gene silencing, which is a process that they use to silence genes, but it's only temporary and it can't be passed on to offspring, while my research focuses on a way that we can pass on those edits and those traits to offspring. So once it's in the TRV vector, I can then transform it into agrobacterium. And agrobacterium is used to infiltrate the plant, pretty much infect the plant with my guide RNA that I designed. So it's quite literally taking a syringe of the solution and infiltrating the leaf. So injecting the leaf with the solution. And as you can see by this smaller image over here, the FtRNA is actually moving the guide RNA into the meristem. So the meristem is where all the action takes place and the replication and new cells are formed. So FtRNA is a mobile guide RNA that induces flowering. So by moving the guide RNA into the flowers, which will become seeds, that's what's essentially causing the progeny to inherit the gene edits. And now if you move on to the results, which is what I've been working on the bulk of the summer, is collecting this data. So once my infiltrated plants had produced seeds, I grew the plants and I sowed the seeds, and I wanted a big sample size, so I did around 50 plants for each transcription factor, so that was about 100 plants. So a lot of plants, and I had to extract DNA from each of them, so it took a long while. But my preliminary data here in figure 3 definitely is a good indication that some mutation took place. Because if you look at the wild type sequence, a little past the guide RNA, it's pretty normal, pretty straightforward, no changes. But if you look at the mutant sequence, there seems to be a bunch of discrepancies in this area, a little past the guide RNA, so where the mutation took place. And that's an indication that something went wrong, that there was a mutation, and that's good for me. But just to make sure that this isn't a cause of some other weird thing that went on, we're going to, let me backtrack a little bit. Actually, the Costfield Bintamiana is tetraploid, so there's two sets of the same genes, meaning this discrepancy can be caused by the two genes running into each other when they're being sequenced. And to rule out that possibility, going forward in the fall, I plan to continue this research. I haven't been able to do it over the summer, but I plan to clone these genes into vectors that I can then sequence separately. And I can then compare it back to the wild type sequence. And I can pretty much see, are there discrepancies or are they the same? And if there are discrepancies, then a mutation took place and my process that I've been working on works. And then after that, I can then do an aphid bioassay, which means I take aphids and I place them on the plants that have the mutations and pretty much see how do they survive, how do they reproduce, how are they changes when compared to a wild type or a control. And then I can link that back to the infector protein and see whether or not that was increasing plant defense or reproduction or decreasing plant defense or reproduction. So that's what my research has really been focusing on.
All right, that's fantastic.
Thanks.
That's a really clever idea of injecting it into the leaves so it gets into the meristem. Usually when you hear, people use like tissue culture, plant tissue culture to create new transformed plants. Was this your idea, or do other people doing this right now? Or
this is actually a pretty new technique. So I was basing this off of some previous paper and literature that I studied up on, and pretty much using the same techniques, but with my topic in particular.
Gotcha.
Yeah.
Oh, that's fantastic. What was your favorite part of being here at BTI this summer?
Probably my favorite part was that's a tough one. I would say just getting to know the other people in the lab. Definitely during the semester and given COVID, it was harder to interact. But now that I was here full day, mask restrictions were lifted during most of the summer. Yep. It was a good experience to get to know people, learn what they were doing, which was really interesting. Yeah.
All right. Well, that's fantastic. And you mentioned you're going to continue this on in the fall. Do you think you will continue on doing plant science in your career?
I'm actually a plant science major here at Cornell.
There you go.
So I definitely plan to do something with plants, but definitely this experience has made me realize that I like working with plants and like being out there. So maybe my research interests may change, but I definitely want to stay working with plants.
That's great. Well, thank you so much, Miguel. That was fantastic.
Thank you.
Cheers. And on to, we have one more before our next break. And I will move here. That's okay.
Thanks, Manny.
Are you Olga?
Yes, I am.
If you want to stand on this side, I think this might be the better way.
All right. Perfect. All right.
All right.
Thank you.
I'm now here with Olga Kilnitsky. Did I pronounce that correctly?
Melnitsky, but that's okay. It's okay. It's a mouthful.
Thank you.
Yes.
So could you tell us a little bit about where you're from and whose lab you're in and what your research project was this summer?
So I'm from Richmond Hill, Queens. I'm a student at Cornell University, and I was working in Magda Julkowska's lab, the stress architecture and RNA biology lab. So yes, and my research is mostly focused on abiotic stress and plant responses to abiotic stress and mechanisms, things like that. So I should start the presentation. Yeah.
Yep, sure.
So I wanted to start my presentation by having you guys envision a scenario. So it's the peak of summer. You're in the middle of an endless field, and you only have one water bottle, and it rains once every couple of weeks. What would you do to survive? While this may be a mere thought experiment for us, this is reality for about 60% of the legumes grown worldwide. And this is what motivated me to spend my summer researching and developing methods to assess the tools that these legumes have to overcome and to thrive in drought stress. So for my experiment, I chose cowpea and tepary beans, which are both native to arid regions of Africa and southwestern US, respectively, as my drought tolerant crops. And I chose common bean as my drought sensitive crop, which I'll be using for comparisons. Half of my plants were kept at 60% water holding capacity, while the other half were kept at 10% water holding capacity to simulate drought. These watering conditions were maintained by a system that I developed using an Arduino and Raspberry Pi. The system automatically weighs the plant pots and then waters them to a target weight and then weighs them again. That data is then exported into a Raspberry Pi computer, and then it can be further analyzed for things such as daily water use. To assess photosynthetic efficiency, I used the PhotoSync-Q multi-spec device. Once stress was initiated, I took measurements of the first true leaves once every day for every plant, and the device provided me with measurements such as relative chlorophyll content, leaf temperature, and overall and quantum yield of photosystem 2. I also was very lucky during my summer internship to help on developing a high throughput phenotyping image theater. However, the complex growth patterns of legumes make it very complicated to accurately image them, and they pose a lot of challenges. The current setup we have is a Raspberry Pi camera will take images of the legumes as they rotate on a platform. These images are then exported to Plant CV and analyzed for things such as overall plant shape, plant area, branching patterns, and things like that. So here we see within the first week a significant decrease in daily water use across all species that were grown in drought. However, this decrease was most substantial in common bean and cultivated tepary beans, while the more drought-tolerant cowpea and wild tepary showed more economic water use from the very beginning, from the very get-go, even at 60%. And this result is, thus allowing them to adapt much faster to decreased water availability. And this result is backed up by their slighter decrease in water use as well as their higher leaf temperatures. Traits reflecting photosynthetic performance, such as chlorophyll content and quantum yield, showed great variation where wild tepary actually performed the poorest compared to cultivated species, while cowpea performed just as well as cultivated species while also using less water. And this result specifically highlights the fact that there's a possibility for different drought tolerance mechanisms present across species, across genotypes, and that's something that is very promising to look into. Upcoming studies in this topic specifically include optimizing the watering system and the imaging system, and further down the line, we envision ourselves looking into phytohormone levels, root and shoot system architecture, as well as ultimately looking at natural variation and heritability of these traits. Thank you for listening to my presentation.
Yeah, that was fantastic. That result there was a surprise. I would have expected the two drought tolerant to be paired up and the other two to be not paired up. But that, like you said, that goes to different mechanisms, possibly.
Yeah, for sure, and especially because they're in different genuses. It'll be interesting down the line to measure something that's in the same genus as cowpea, and see are those mechanisms conserved within the genus. And then also we have 5 research grade genotypes of cowpea, which we're also going to be looking into to see how those traits vary across the genotypes.
So will you be continuing this research yourself or handing it off?
Yes, because I'm a student at Cornell, I'm lucky enough to be able to stay in this lab and stay with my research mentor, which is going to be really exciting. A lot of future things. This is definitely a project from the sprouts, and once we have developed methods, it's going to be really interesting to see what we do further on to develop those traits, to see those traits and how they respond.
Yeah, that's fantastic.
Thank you.
What was your favorite part of the summer internship program?
Oh, of the summer internship program, I would say it would be just, working with my mentor, getting to know new people, and being with a lot of other people who are also interested in the same thing that you're interested in, but also they're doing things so different than what you're doing, and learning things about that. It's just been amazing. I personally love just being in a community like this, of a scientific community that is so supportive. Yeah.
And that's great. We're glad you were here.
Thank you.
That was a fantastic presentation.
So
Good job.
Thank you.
Lucio Malbas? All right. Hello and welcome back to Boyce Thompson Institute at the George and Helen Kohut Symposium. I'm here with Kieran Lucio Malbas. And could you please tell us about where you're from and what your research project was this summer?
Yeah, so I'm from here. I'm from Ithaca, New York. And so my research project was, should I just hold?
Oh, that's fine.
So I was looking into cardiac glycosides, which are a type of defensive metabolite found in various plant families. And specifically, I was looking into one type of cardiac glycosides, which are cardenolides, that is found in the genus Erysimum, which you can see here. And this is just an image of the general structure of a cardiac glycoside. And so cardiac glycosides work by inhibiting the sodium-potassium pump. And this is how they can defend in plants if they're ingested by insects. They will negatively affect the insects, or they should. So we were looking at the mutant 635 line of Erysimum charanthoides, which differs from wild type in that it produces less cardenolides in the wild type plants. You can see that here. So we were wondering how this lower cardenolide content would affect insect herbivores. And specifically, we looked at peach aphids and 2 types of caterpillars. So for my choice assays, I placed 10 peach aphids at the intersection of a wild type leaf and a mutant 635 Erysimum leaf. And then I left them per day to just wander to either leaf and presumably consume the leaf that they had a preference for, and then counted the number of aphids on each leaf after 24 hours. For the caterpillars, I did something similar, but I only used 2 to 3 caterpillars, and I left them for a bit longer because caterpillars have less mobility than aphids. And then I used this software called LeafBite, which basically lets you fill in where the edge of the leaf was, and then it'll tell you about how much was consumed by percent and a number of square centimeters. And then for the performance assays, we were also looking at how this difference in cardenolide content would affect reproduction in aphids. So I put 3 aphids on a full mutant 635 or wild type plant, and I caged the whole plant in a plastic bag to keep the aphids on it. And then I left them for 2 days and looked at the number of aphid progeny. So in all experiments but the first aphid choice assay, we found more either reproduction or more consumption of the mutant 635 leaves. Although on the second aphid choice assay and the caterpillar choice assay, these results weren't statistically significant. However, on the first aphid choice assay, we found the opposite result, which is pretty unexpected. which is that the leaves with more cardinalides actually had more aphids feeding on them, and this was a statistically significant result. So this shows that the mutant 635 line definitely has an impact on aphid, on insect preference in general, but it's not as clear and distinct as would be expected. So this could be due to a number of things, possibly including some physiological difference in different types of aphids or between aphids and caterpillars. And there could also be a difference between the 635 mutant line and wild type Erysimum that isn't known besides just their differing cardinalide content, because we don't really know exactly what genes are linked to cardinalide production specifically, or what pathway biosynthesis of cardinalides takes. So this could benefit from a lot more research into the genetics of it, pretty much. And this can be beneficial because cardiac glycosides are used as heart medications, so that has a medical application. And of course, by understanding the pathways for cardinalide production in Erysimum, this could be applied to other plants, like possibly crops, and that would mean we would need less pesticides, and we could also make more pest-resistant crops.
That's a really great project. What was your favorite part of doing that this summer?
I would say my favorite specific thing we did, I think, was attempted transformations of Erysimum flowers using agrobacteria. So that was really exciting because I guess, well, first of all, we got to use a vacuum machine, which was pretty cool. But also, I think that would be, I think that was the thing that represented, I guess, the culmination of a lot of stuff we'd been working on earlier. And I mean, I won't really, I won't get to see how that works out personally because it'll take a couple months. But it seems like it would, if it works, it seems like it could have really cool, it would be really cool to see how that works out. Yeah.
Right. Well, that sounds great. That was a great presentation. And thank you so much for being here this summer. All right, congratulations. Okay, on to our next poster. Okay. Okay. Hey, pardon?
You can listen as well. So I'm gonna just, I think, restart.
Pretty much, yeah.
Okay,
Yeah.
perfect. Hi, my name's.
We have a thumbs up. All right. I'm here with Katie Eisenhut. Did I pronounce that correctly? Great. Thank you. Could you tell us where you're from and what your project was this summer?
Hi, my name's Katie Eisenhut. I'm from Ithaca, New York, and this summer I researched the antibacterial properties of kombucha against Candida Liberibacter asiaticus in search for an organic solution to address citrus greening.
Great. Could you tell us more about your research?
So Candida Liberibacter asiaticus, also referred to as CLAS, is a gram-negative bacterial pathogen responsible for the citrus disease Huang Long Bing, also known as citrus greening. Spread among trees by its insect vector, the Asian citrus psyllid, citrus disease or citrus greening has become one of the most harmful citrus diseases in the world. Chemical expenses, environmental concerns, along with increasing fear regarding the development of chemical resistance within the Asian citrus psyllid population has led the citrus community to search for more organic solutions. So one possible organic solution would be kombucha tea formulations that growers could make or buy themselves. Our interest in kombucha stemmed from studies indicating that kombucha had antibacterial properties against both gram-positive and gram-negative bacteria under a variety of different pH and temperature conditions. So there were two aims to my experiment. So because CLAS is actually not culturable, my first aim focused on the cultivable relative Liberibacter crescens, along with another phytopathogenic citrus bacteria known as Xanthomonas citri, in which we did in vitro biological studies. I hypothesized that L. crescens and X. citri would experience an inhibition in growth when exposed to kombucha. For the second aim of my project, we looked at the relationship between kombucha and CLAS through detached leaf assays, and I hypothesized that what the CLAS is exposed to kombucha via detached leaf assay, there would be an inhibition in growth. And so while I had no statistical data for my first aim, we can see through these images that there's at least some inhibitory properties of the kombucha between T0 and T1. And so for the second component of my project, where we were with the detached leaf assays, we can see in this graph I had my 3 treatment groups. But through my t-tests and other analytical processes, we concluded that CLAS was not inhibited by the kombucha. More data could be conducted and further replication of the study should be done. However, kombucha may not be a useful disease mitigation strategy to address citrus greening, but there is a lot of studies indicating that it has a significant impact on the human microbiome. And so if we can understand the antimicrobial or antibacterial properties of kombucha, we can transition this knowledge into the citrus sector. So that's a brief intro of what it is.
All right, that's fascinating. I never would have thought of trying kombucha to spray citrus trees. That's a really creative idea. What was your favorite part of doing your BTI research this summer?
Just generally, I learned a lot. My mentor, Dr. Heck and Dr. Higgins, were extremely helpful. This was my first experience, so broadening my knowledge in microbiology and some wet lab techniques was really, really nice.
That's fantastic. Do you think this will impact what you choose to do for your career?
I think, yeah, for sure. I mean, I'm a food science major, and so looking at food microbiology and gaining this experience, I think, is extremely valuable, and I can take it with me to Penn State.
Yeah, that was fantastic. That was a great presentation.
Thank you very much.
All right, thank you.
Thank you.
Right here we have latifolia, so you can
We'll move on.
just from here that there's a big difference in the
Oh, Jesse?
Yes.
Hey, how you doing?
Hello.
Sorry to interrupt. He's up for the live stream. Are you all set?
Yeah, almost set to go.
All right.
Sorry, Shirley.
I'm gonna sneak over here.
Hello. So
One second. Make sure the camera. All right, we're good to go. I'm here with Jesse Derby, and could you please let us know where you're from and tell us a little bit about your research, whose lab you're in, and what you do?
All right. I am from Larchmont, New York, so not too far south of here. I am in Anurag Agrawal's lab, so right here. It's within the Department of Ecology and Evolutionary Biology, and the lab focuses a lot on milkweeds and cardenolides, and basically the interaction between plant toxins and then insect physiology. So it studies a lot on co-evolutionary mechanisms within plant-insect interactions. So particular focus of my project over the summer was studying these class of natural toxins called cardenolides. So cardenolides are found very prevalently within milkweed species, and they target a particular enzyme, Na+, K+ ATPase, on animal cell membranes, and they inhibit that activity. And so they're used basically for plants' chemical defense and resistance against insects. So cardenolide mixtures, there's a good variety of them. Within some species, there can be up to 30 distinct compounds of cardenolides. And so understanding this diversity is really crucial to plant chemical defense and understanding those mechanisms. And a particular interest to the study was that there can be variation even within a species across the tissues. So seeds and leaves and roots will all have different mixtures of cardenolides and different concentrations and types. And so the main thing we were studying is we were trying to find for Asclepias syriaca, which is common milkweed, you find it very prevalently in the wild in the Northeast. Are there these differences in concentration and type? And then do differences in cardenolide mixtures create different inhibition effects, create different effects on the insects feeding on them? And are these effects driven by the concentration of the cardenolide or the potency of the cardenolide, or both? And so during very basic methodology, grew and collect plant samples, analyze them for HPLC to identify and quantify their cardenolides within. And then at testing for inhibition, we ran sodium pump assays on resistant and then non-resistant type enzymes, where we basically put in the cardenolide mixture ATP, the enzyme that it inhibited in a reaction buffer, and tested for the amount of activity by seeing the amount of phosphate that was cleaved off by the enzyme. And so going into the results a bit, we found pretty clear variations both in concentration and type for cardenolide mixtures within common milkweed. So even just looking at the scale of the graphs here, you can see the pretty stark differences in concentration between, especially like the seeds compared to leaves and roots. This is the scale of the graph, shows a very clear difference in concentration. And then also in the type. So we see these first 5 cardenolides here are expressed pretty readily within the seeds, but they're basically not expressed at all within leaves and roots. And you find examples of this throughout the spectrum of retention times. So the first question we were trying to address was answered pretty much in the affirmative. And so getting into differences in inhibition, this is kind of a secondary part of the study. We tested 10 milkweed species on doing those sodium pump assays, and we found pretty clear differences. So right here are the IC50 points, and IC50 point is basically just the concentration at which there's 50% inhibition. And so you can see right here the differences in concentration. So this is for Pultra and Latifolia, which are 2 of the species here. This is on the non-resistant enzyme. This is on the wild type fly. There's a difference in the IC50 point. So we saw that cardenolide mixtures create different effects in inhibition. So going into it, is that difference concentration or potency based? We found that you can see right here, the concentration of the cardenolides varied pretty wildly across the 10 species. And this is also looked into here. This is the amount of seed tissue that was required to reach that IC50 point, that inhibition. So you can see, obviously, there's a big difference across the resistant and the non-resistant, right? But even across the same type of enzyme, you'll see pretty clear differences in the concentrations required to create this IC50 point. And then, so there seemed to be an aspect of concentration. And then going into here, this is basically the amount of like pure cardenolide required to reach IC50. It's basically a measure of the potency of the cardenolides, how much is required to inhibit. And we see pretty clear differences in the potency of the cardenolides as well. So not only do we see this difference in inhibition, it seems to be driven by both concentration and potency. And so that's basically summing that up in the conclusion right there. And then the further research is to basically ask, so we saw this variation, we know that it has an effect, but why is this variation? So the further research is basically in specialized defense. So we think that one reason that these plants might have these differences is because there's different specialist herbivores that eat on only particular parts of the plant. So we believe that these cardenolide mixtures might be adapted to specifically target the insects that feed on the particular parts of the plant. And so the next steps are really to go into that direction, focus on the insect interactions, and to test the inhibition of, let's say, like a specialist feeder on seed insects versus a specialist feeder on leaf insects, and see which suffer more from inhibition of seeds. And that's a direction to take it in.
Is that something you will be doing, or will you be handing it off to someone?
I think that it will be handed off to the lab. I'm hoping to return over the academic year. We'll see if schedule permits it. But if not carried out by me, it will be carried out by the excellent members of my lab. So
excellent. What was your favorite part of the BTI internship program?
I think my favorite part was just getting to work with my mentors. So I don't have to mention other, but Amy Hastings and then Soze Goldar. They were both excellent mentors, and they taught me through every part of the process, basically.
Oh, that's fantastic. Well, you did a great job. Thank you so much. Hello. What's that? Okay. We are moving on to Zoe James. Hello, Zoe. Could you please tell us where you're from and what your research project was?
Yeah, I'm from Ithaca, and my project was in looking at the DUF247 mutant and examining it in salt stress and seeing what part it plays in salt stress response in Arabidopsis.
Great. Could you tell us more about it?
Yeah, so soil salinity is a major threat to agriculture right now and reduces plant productivity. So right now, it is really important to increase that and look at plant architecture and see how it is affected by the soil salinity. So in a previous GWAS, it was identified that the DUF247 was a loci that could potentially play a role in the root to shoot ratio in Arabidopsis. So, and for my project, it was to identify the potential role that this gene has in contributing to regulation of salt stress response in Arabidopsis. So we had three experiments. The first one was a germination experiment where we germinated a DUF mutant and a Columbia wild type of the Arabidopsis on media. One was half with salt and then the other as control. And after 4 replicates, there was no significant difference between the two, except for one. And so we plan to repeat this experiment with different components of the media. Our second experiment was a survival where we germinated the seedlings on control plates and then transferred them to plus or minus salt treatment, where we again saw there was no significant difference between the Columbia and the salt, the Columbia and the DUF. So finally, our third experiment was in soil, as the first 2 were on media. So we had 2 trays of individualized seedlings of the Columbia and the DUF, where we gave 1 tray a salt treatment and the other one was left as a control. And these trays were placed into rigs that were equipped with an auto imaging camera that took a picture every 30 minutes. So these pictures will be imported into PlantCV, which is a computer program that can measure the area of the rosettes. And that way we'll be able to compare the above ground architecture of the 2 genotypes and see how soil salinity affects that.
Well, that's fantastic. And was that a lot of fun working with those rigs this summer?
Yeah, definitely. It was so important to just see everything, and I'm really excited to in the future conduct more research.
Do you plan to continue on when you go to college doing more research?
Yeah, I hope so.
Oh, that's great. Well, this was a fantastic presentation. You did some great work this summer. All right, thank you very much. I'm here with Michelle Hassan, and could you please tell us where you're from and what your research project was this summer?
I'm from New York City, and my research project was based on a disease in bees called Crithidia. So yeah, my research this summer was based on bees and more specifically disease in bees. And the disease that we focus on is called Crithidia. And so we know that Crithidia can specifically infect bumblebees. And so that's why we always put the genus of the bumblebee, which is Bombi, next to Crithidia. So we always say Crithidia bombi. So because we know that Crithidia can infect specifically bumblebees, what we want to find out this summer is whether or not Crithidia can infect other bee species. So we know that bees can be infected through the feces that other bees leave on flowers when they go out to pollinate. And so there were 2 methods that we used to determine whether or not other bees can be infected. And one of them was observational, the other was experimental. So for the observational part, what we did was we went out and we collected bees from the wild, from different locations in Ithaca, such as Ithaca Children's Garden or the Cornell Botanical Garden or Van Horn Park by downtown Ithaca. And what we did was we went out with nets and we caught the bees with our nets, and then we took a tube and we put them in that tube, and then we brought them back to the lab. And once we brought them back to the lab, what we did was we dissected the bees. So we used forceps to take out the guts from the abdomen of the bees, and then we looked at and then we put the guts into a vial of PBS solution. PBS is phosphate buffer saline, and so what it does is it maintains the pH of the guts and the PBS. And so after we put the guts and the PBS into a solution, we waited 2 hours and we put that solution onto a hemocytometer and we looked at it under a microscope. So if we knew that that those guts had Crithidia, they would look like this. And basically, Crithidia is a very teardrop-shaped parasite. And also, by the way that it moves, it's very elongated when it moves. So that's how we knew it would be Crithidia. And so this is the data that we got from the wild bee screenings. We separated it into two tables. One is by family and one is by genus. So out of the 5 families of the bees that we sampled from, we found that each one had positive samples except for Collectidae. Collectidae had no positive samples for Crithidia. And as for the genuses, we collected bees from 24 genuses, and 10 out of those genuses were positive for Crithidia. And something interesting that we found is that the bombus, or bumblebee species, we collected 48 of the bumblebees, and only 2% of them were positive. But what we found interesting is that the mean number of Crithidia cells that were positive was very high compared to the rest of the bee genuses, and so that just shows how prevalent Crithidia can be in bumblebee species. And so, as for the experimental part, what we did was we inoculated a certain amount of the wild bees that we caught. So what that means is we intentionally made them sick. We fed them with Crithidia infected bee guts from a colony that we already had in the lab, and we mixed it with sucrose so that the bees would find it tasteful. And then we waited 1 week and we dissected them to see if they had Crithidia or not. And so this is what we found. So we then again separated the tables by bee family and bee genus. And so the red dotted line, the dashed line, shows the threshold for replication. So what we found is that the Apidae and Collectidae families had very high replication amounts. And as for the bee genuses, Ceratina and Hylaeus had very high replication. And so basically for next steps, what we found is that not only bumblebees can be infected by Crithidia, there's also a lot more bee genuses that can be infected. And so now that we know that not only bumblebees can be infected by Crithidia, what we want to find out is how location or environment can affect certain bee species, and whether or not that affects how they can get infected or how prevalent Crithidia can be in those bees. And another thing that we found interesting is that Hylaeus, this bee species, we found that none of the Hylaeus that we sampled had Crithidia, but when we did the inoculations, it had very high replication. So something else that we want to find out is why Hylaeus in the wild had no Crithidia, but they're very prone to it. Yeah.
That's fascinating. And what was your favorite part of the program this summer?
I would say my favorite part was going out into the field with my lab. It was a lot of fun catching bees with my lab members. Yeah.
Well, that was great. Thank you so much for sharing your research with us. That was really fascinating. Okay, have a good rest of your summer. Okay, we will move on. Hello. You're Linda?
Yeah.
All right. We have a thumbs up. That means we're good to go. Linda Lee?
Yep.
Thank you for presenting with us today. Could you please tell us whose lab you're in, where you're from, and what your research you did this summer?
I am working in Dr. Eric Richards' lab. I'm from Woodbury, Minnesota, and I study bioengineering at Cornell. Was I supposed to say anything else?
Just can you tell us about your research?
All right, do you want me to hold this?
If you like to.
Okay,
That's fine.
Okay, so we were exploring the physical characteristics of plant nuclei this summer, specifically in tomato. So the nucleus is an integral part of the cell to protect the genome, and we want to see how the nuclear architecture worked and how the cells will respond to physical forces, because that's not really studied yet. And this would help lead to future research in mechanosensing. And that's when how cells respond to physical forces and outside. For example, how they change their gene expression to adapt to their surroundings. So the morphology of nuclei is based mostly on environmental factors and cell type and function. For example, in animals, you can have cells in the immune response they have like more pliable nuclei so that they can squeeze through small channels in your body to get through sites that are infected and to fight off like viruses and bacteria that are just there. So our first step was to prepare the nuclei for studying. And because in animal cells, you can just directly grow the nuclei on, or the animal cells on dishes, and then poke the nuclei through the cell membrane. That's like really easy to study, and they've already done that a bit in the biomedical engineering labs. But we have to chop it up manually to get through the cell wall, and then use non-ionic detergent, Triton X100, to lyse all the chloroplasts and all the other remaining organelles and junk. So as you can see here, we tried a bunch of different concentrations, and we wanted to make sure we had a lot of nuclei. This was like very few compared to more nuclei over here, but more debris. So we settled on the second image, just like an in-between of less debris, more nuclei. And then later on, we tried other methods to see if we could get more nuclei, less debris. And it didn't really work out. As you can see here, it's like full of junk. And we decided to go back to our simple method of chopping in detergent. All right. And finally, we were able to plate the nuclei onto microscope slides and petri dishes. I don't know if you can see it through the camera, but we would like to, in the future, work with the Lamerding lab in biomedical engineering to use a nano indenter on an inverted microscope to squish nuclei, basically, and calculate the Young's modulus and storage and loss moduli and see basically how rigid they are. The second half of my project was to explore the nuclear phenotypes caused by mutation in the NMCP2 gene. So this gene encodes for nuclear lamina protein that help maintain the structure of the nuclei. And if you change some of the amino acids in the N terminal, then the plant still keeps it's still alive, but the phenotypes of the nuclei change. And as you can see in these pictures, on the left side is the wild type, and the nuclei are like football shaped, they're really long, kind of squishy. And on the right side, they are more round, they're mutated. So specifically, we wanted to look at some parameters like area of the nuclei, and these are guard cells in the stomata. And we found that the mutants actually had smaller nuclei in the guard cells. Next over here, we found that the mutants had more round nuclei, and they weren't as oval shaped anymore. And right below that, we measured nuclear positionings to see if they were actually kind of across from each other in the gut cell. And I don't know if it's easy to see, but I measured the long side, see if they were like directly across from each other. We found that the mutants were more crooked, and it could be because the nuclei are smaller and they squished around more, or because there's some sort of thing in the mutation that affects the cytoskeleton, and it just is no longer fixed, and it just goes around, you know. Our next steps from this project is to extend our studies to Arabidopsis to see if there are any changes between plant species, as well as changing the epigenetics, such as methylation of the DNA, or changing heterochromatin to euchromatin ratio. And like by condensing the nuclei or condensing the DNA, we can see if the nuclei changes size or rigidity, stuff like that. You have any questions?
Yeah, that was a really cool project. It sounds like you got a lot done this summer.
Yeah, thank you.
What was your favorite part of the program this summer?
I actually really enjoyed learning about microscopy. I didn't expect to be doing that going into the program, and I learned how to use like the episcope, confocal microscope, and it's like so pretty as well. It's like really satisfying.
Yeah, we got a lot of neat microscopes here.
Yeah.
Well, thank you so much for sharing your research with us. It was really cool.
Yeah, thank you.
All right, have a good rest of your summer.
You too.
Bye-bye. We will move on. Okay. Hello. You're Eddie? All right. Nice to meet you.
Nice to meet you. Want to get started?
Yeah, we're good to go. Can you tell us whose lab you're in and where you're from and what your research was?
Yeah, sure. So I'm Eddie Almanza. I was in the Frank lab. I'm from North Carolina, and my research or my project was focused on trying to uncover genetic loci underlying fruit development in eggplants. So should I just go and get started?
Yeah, absolutely.
All right, awesome. So eggplant is grown around the world, and it's really important economically and culturally. And it's thought to have been domesticated in Southeast Asia. And the goal of our study was to identify candidate loci and genes controlling important fruit traits such as width, weight, length, and shape. So to do this, we grew 100 accessions in the Guterman greenhouse here at Cornell. And these include wild, semi-domesticated, and domesticated eggplants. And this map here shows where we got each of the accessions. And the dots that are purple are domesticated eggplants, yellow is semi-domesticated, and green is wild. So we then performed short read sequencing on these accessions, and we aligned the resulting reads to the reference genome recently created by my mentor, Hamid. And once that resulted in a database of single nucleotide polymorphisms, SNPs, that showed the genetic variation between accessions. And then we were able to perform genome-wide association studies, GWAS, to try to discover loci controlling these traits. So if you look at this phylogeny right here, the accessions in green are wild, the accessions in yellow are semi-domesticated, and the purple ones are domesticated. And there's two main takeaways from this. One is that there was clear selection for larger fruit during domestication, because these domesticated fruit are much larger than the wild and semi-domesticated ones. And another thing we noticed was that there are two independent domestication events, or there may be. One that occurred here, and then one that occurred here. And then the box plots you see here just kind of help strengthen the argument that there was selection for larger fruit, because as you can tell, the domesticated fruit, which are still in purple, are heavier, longer, and wider. So we then performed a principal component analysis, which we use using the genomic data. And that just shows the variation or the genetic variation and genetic distance between all the accessions. So if it's clustered close together, that means it's pretty close or related, pretty similar. And if it's spread out, it just means it's genetically, what's it called, different. There's a lot of genetic variation. So the wild accessions are spread out, which means there's a lot of genetic variation, which is what you'd expect. And the purple ones, which are domesticated, are close together, which means there's low genetic diversity between them. So these are the results from our GWAS. These are Manhattan plots, and each peak you see is a location on a chromosome that might be associated with that trait. So as you can see here, there's peaks on chromosome 2, 4, 8, 9, and 10, especially on chromosome 9. That's a really prominent peak. But that just means that there are loci on that chromosome, or within the genome really, that are probably linked to that trait that you're looking for. So curved fruit, looks like there are loci on chromosomes 4, 9, and 8, 9, and 12, especially chromosome 12. And for elongated fruit, the peaks weren't as prominent because this is a cutoff that shows, or if it's below that cutoff, it's not statistically significant. So there are two places within the genome, chromosome 2 and chromosome 11, that might be linked to it, but we need to do more research just to make sure. So yeah, and to sum that all up, this just proved that there was selection for larger fruit during domestication. And there might have been two separate domestication events for eggplants, which is also very interesting. And we discovered multiple novel loci, or multiple genetic loci for eggplant improvement, especially within the fruit shape. And the GWAS results indicate that fruit shape might be controlled by multiple genes on different chromosomes instead of just a single gene. So,
yeah. That was fascinating. That was really cool. Yeah, could you tell us what your favorite part of the program was this summer?
So I've never done research before, so I don't want to be cheesy and say the whole thing was my favorite part, but I learned so much the entire time, and I really enjoyed it.
That's great. What is your major?
I'm a plant sciences major, so fitting.
Yeah, that's great. And I have to say, from a comms perspective, I love this poster. It is so colorful and engaging. So you did a great poster. I love it.
Leave the words out.
What's that?
I try to leave the words out and just let the pictures do the talking.
Yeah, they do. They really make you want to come right in and take a look at it.
Awesome.
So great job on the poster too.
Thank you so much.
All right. Well, have a good rest of your summer. Okay. We'll move on to the next person. Hello.
Hi there.
I move to this side. And you're Abby?
Yep.
All right, nice to meet you.
Nice to meet you.
We're good to go. Abby Kelly?
Yep.
Could you please tell us what lab you are in and where you're from and what your research was this summer?
Okay, I am from the Jender lab, or I come from Western New York, and this summer I've been working on an assessment of benzoxazinoid content in maize leaves. Should I take the mic?
Yeah, if you like.
Okay.
Just
So-
keep it up right up there. Yep.
Hi there, my name is Abby and I work in the Jender lab. So this summer I have been working to search for differences in the wax content of different lines of maize. More specifically, I have been looking for differences in the wax benzoxazinoid content. Benzoxazinoids, or BXs for short, are secondary metabolites that contribute to insect resistance in maize. The cuticular waxes of the leaves are the first physical line of defense that plants have against insect herbivory, so their BX content could play a really important role in determining insect resistance in plants. So the first part of my work this summer involved developing and optimizing a procedure to actually extract the cuticular waxes from the leaves. So our finalized procedure involved dipping a leaf section into chloroform to remove the waxes, and then drawing that chloroform off under a nitrogen stream to kind of preserve the BXs and make sure they didn't oxidize. So after I had completed the procedure and I had finalized the procedure, I did an analysis of the NAM founders. So the NAM founders are just a whole set of different lines of corn that are very genetically diverse. So they include B73, which is my baseline for comparison. And I also analyzed a BX1 knockout mutant, which is the gene that I have highlighted in blue right here. So BX1 is a gene in the benzoxazinoid biosynthesis pathway. And when BX1 is knocked out, I should not be seeing any of these downstream BXs. So after I analyzed the NAM founders, I selected the 4 most extreme lines to look at in further detail, the 2 lowest, the 2 highest. I re-extracted waxes from these lines, and I reanalyzed them again via liquid chromatography mass spectrometry. So this bar chart here is just kind of showing an example of what we were looking at. So this is H-dimboa glucoside, which is one of the BXs in the BX biosynthesis pathway. The B73 comparison I have highlighted in green here. And then these are the different lines that we were comparing to B73. We used a Dunnett's t-test to determine whether the differences we saw were statistically significant. And the red stars there indicate that we did find a statistically significant difference. So this heat map that I have over here is kind of a summary of our findings for the summer. We have the different corn lines that we were looking at over here, and we have the different BXs along the top. So the stars indicate the level of statistical significance, and the colors indicate whether they were lower than B73 or higher than B73. And again, B73 is just our baseline for comparison. So in the future, there are a bunch of different steps we can take. We'll probably be performing insect bioassays to determine, they'll just help us determine the practical effects of the BXs and the waxes. We could spike artificial diets. We're not exactly sure how that would work yet. We also may perform a GWAS, which is a genome-wide association study, and that would help us determine, for example, which genes are helping transport the BXs into the wax. We also might do a procedure to extract the epicuticular waxes as opposed to the total intracuticular waxes and compare those. So big thanks to Cornell and BTI for giving me this opportunity.
Well, that was a great presentation. Thank you so much for sharing that with us. What was your favorite part of the program this summer?
I think just getting a lot of experience working in the lab and on data analysis. So far, most of my work has been wet work, but this summer I got to do a lot with data analysis because we used the LC so much. So it was really nice to get that experience.
That's great. Do you think this project will affect what you do in your career?
Absolutely, yeah. I've been working at BTI for almost 2 years now, and this project and just all of my work here has definitely made me want to keep going in plant science, which I didn't think I would want to do when I started undergrad.
Well, that's fantastic. Thank you so much for sharing your research with us.
Thank you.
All right, have a good rest of your summer.
You too.
And now we will move on. I think we're over here with Lily. All right, I will sneak over to this side, and we are here with Lily Yang. And could you tell us where you're from and whose lab you're in and what your research was this summer?
Yeah, my name is Lily, and I
live in Ithaca, New York, and I've been working in the BCBC with Dr. Susie Strickler. And my project over the summer was using comparative geonomics to study Asclepius. And so Asclepius has been a popular study subject for biochemical, ecological, and medicinal purposes, specifically Asclepius syriaca, which is common milkweed, and Asclepius cursavica, which is tropical milkweed. However, they lack geonomic tools. So BTI has developed and generated new Asclepius assembly, and I have been used in order to improve existing resources. And I've been using the new resource, like the new generated assembly, in order to compare it with previous studies, and also new other gene species and gene families. And so my work has been into like 3 different components. My first thing was comparing BTI syriaca genome with the published version. And the published version was based on short read data and highly fragmented, whereas the BTI syriaca genome was based on long read data. And so we want to see the difference between short read data and long read data. And I ran a tool called NukeMer, which was a DNA sequence alignment tool. And I was able to find the percent coverage between the two genomes, and it was 11.13%. And it shows that the high coverage long read sequence has greatly been improved and has greatly allowed our ability to assemble a majority of the syriaca genome. And my next thing I did was comparing the syriaca assembly to the cursavica assembly, and these are all from BTI. And what we were interested in seeing was the difference between assembly anchored to a genetic map and one that was not. And in addition to that, also to see the relationship between the two and see if they're syntenic or collinear. And this was my synteny plot that was developed, that I generated using a tool called SynMap. And through it, we can see that the 2 organisms are mostly syntenic, but you see some gaps right there, which is because of translocations. And we think that's because the cursavica assembly did not have a genetic map. So that's what our future work will be. And my third thing I did was gene family analysis of these 5 different species. And we were interested in seeing which gene families were expanded in the Asclepius lineage, and also if they had functions involved in a unique Asclepius metabolism. And yeah, so in this chart, figure 6, you can see it's a phylogenetic tree created by OrthoFinder. And so it just shows all the accepted species tree. And then I also ran a tool called Cafe in order to get the different values there. And the values in green show expanded the number of expanded gene families, and the number in red shows the number of contracted gene families. And then up here, figure 5 shows several different functional categories of the expanded, and this is the contracted in the Asclepius lineage. So that's pretty much what I've been doing.
Wow, that's a lot of stuff. That was really cool. Congratulations on all that. Could you tell us what your favorite thing was that you did this summer?
My favorite thing, I really enjoyed actually the last project I did, which was a gene family analysis, especially the results I was able to receive. So yeah.
Hey, well, that was fascinating. Thank you so much for sharing your research with us. I appreciate it. Have a good rest of your summer. Okay, and Rohit, I'm going to sneak over on this side of you. And we're here with Rohit Lal. Oh, I'll wait for the thumbs up. All right, we got a thumbs up. Rohit, could you tell us whose lab you were in or who you were working with and what your research was this summer?
So I worked in Dr. Susie Strickler's lab, and over the summer I focused on mining for putative orthologs involved in pattern-triggered immunity and effector-triggered immunity in the Solanaceae family. So essentially, I used Solanum lycopersicum as a reference for finding the putative orthologs in the rest of the Solanaceae, which can be seen here. these are all the species that I mined for orthologs. And basically my process for mining those orthologs starts right here, where I look for the coding sequence, protein, genome, and gene models files, most of which came from Soul Genomics Network and NCBI. And then at the beginning part of my internship involved creating this Python script called analysis.py, which basically took the coding sequence file of Selenium lycopersicum and the protein file of Selenium lycopersicum and the same files for the other Solanaceae, and it basically performed all of these tasks, including extracting the gene of interest from Selenium lycopersicum, blasting it, finding the top match, extracting that, and aligning the top match and the original gene. And basically, it collects all the data from that alignment, and it gives you this tabular output down here. So here you can see, it's kind of hard to see, but there's the original gene, there's a top match, and then the kind of important statistic that I looked at was whether or not this gave true or false. And basically true meant that from when you ran the script and there was the blast output, this top match is the actual putative ortholog for this gene. However, if a false came up, that means that I had to look deeper to try and find a putative ortholog for that gene in Selenium lycopersicum in the other Solanaceae. So what I did was I constructed these SynMaps, which is a tool from Cybers. And basically, using this tool, I can look deeper into these Koji plots. And basically, I look at a flanking gene for a gene that did not find a putative ortholog for the other Solanaceae. And I look at, so this gene right here, for example, is a gene that didn't find a putative ortholog in the other Solanaceae. However, using Synteny, we can see that here, this is fairly aligned with this gene right here. And so we can infer that this might be a putative ortholog for this gene. And so pretty much my results from the mining process are over here. So this is the effector triggered immunity and the pattern triggered immunity. And I for this table, I only handpicked one from each type of like potato, tomato, because I had to fit it on the poster. But here, a true on this table pretty much means that for a specific gene in Selenium lycopersicum, in that other Solanaceae species, there was a putative ortholog found. And pretty much going forward, what I'm going to have to do is I'm going to have to look at each of these SynMaps that I generated and see if that SynMap, I can find a putative ortholog like I've done here for the gene in Selenium lycopersicum that could not find a top match from just running the script. And so once I've done that, I'm going to move forward into looking at the evolution of the genes. And I'm going to start by running an ortho finder, which will basically construct ortho groups for all of the Solanaceae with all the Solanaceae protein files. And then I'm going to be using PopGen analysis tools like PAML so that I can really start getting a better picture of the evolution of these genes in the Solanaceae family.
Wow, that's fascinating. You've done so much work here. This is what, your third summer here now?
It is.
Yeah. So what is your favorite part about doing these internships here at Boyce Thompson Institute?
I think the favorite part for me is like, it's never really easy. Like you always have to learn something new to be able to accomplish something at the end. It's never a straightforward process. And I think that's really fun to like go through and experience.
To work through the puzzles, figure new things out?
Yeah. And you learn so many more new tools that I think will be really useful going forward. Yeah.
Well, that's amazing research. Thank you so much for sharing it with us. Yeah, and we'll see you around the building, I'm sure. Yep. Okay, we're down to our last poster. Hello? We'll wait for the thumbs up.
Okay.
All right, we got the thumbs up. So I'm here with, is it Anna Supe?
Yeah.
All right, very nice to meet you.
Nice to meet you too.
And could you please tell us whose lab you are in and what your research was this summer?
Yeah, so I was working in the Thaler lab this summer in the entomology department, and we looked at how pollen diet influences bumblebee colony development and pollen transfer services.
Great. Could you tell us more about your research?
Yeah, absolutely. So we were looking at the system of bumblebees because of their agricultural importance, how they provide, since they're generalist foragers, they provide like a lot of important pollination services to agricultural crops. And we also know that the diet breadth is constrained, can be constrained by agricultural simplification. And so the diet breadth impacts the colony development, but it doesn't, we don't know how this scales up to influence the pollination services. So we looked at the influence of different pollen diets on bombus impatiens, the common eastern bumblebee, to see how that the effects of diet on colony development scale up to influence pollination services. And we looked at the tomatillo system. Yeah, so we thought that like a more diverse pollen diet would enhance colony development, and that since a diverse diet would enhance colony development, that bees on like a monofloral diet would be less likely to forage for pollen, and that and that polyfloral diet, like a diverse pollen diet, will increase cross-pollination in the tomatillo system. So in order to do this, we created microcolonies, and we gave them each one of two diets. We had a diverse diet, kind of to mimic the heterogeneous landscape in like nature, and then we had a monofloral pollen diet to sort of mimic like a monocultural landscape. And then we measured the colony development by proxy of honey cell weight, and then this is like a honey cell that we measured at the end from each of the microcolonies. So after we developed these microcolonies, we had them undergo foraging trials on the tomatillo, and we measured the foraging bout length and whether bees were foraging for pollen or nectar. And then this is when two bees are foraging on both, which was cool. And then afterwards, after the foraging trials, we removed the stigma and transferred its contents to a slide where we would measure the effect of diet on cross-pollination by looking at the number of opposite grains, digrains deposited, because tomatillo are self-incompatible. So we needed to see like opposite pollen. So basically we found that diet did not have a main effect on the colony end weight, but it did, the longer that the colonies were ascribed to a certain diet, the polyfloral diet would increase over time. We found that there was no effect of diet on the likelihood of bees foraging for pollen, and that diet treatment had no main effect on pollen grain stigma deposit. However, you can see like how the polyfloral diet spent more time foraging compared to the monofloral. So basically we found a lot of unexpected interactions that didn't necessarily match our hypotheses, but that did have really cool implications for future research. So we figured we could do this on a more ecologically realistic spatial scale, as well as over a longer period of time. And then we hope that this research can be used to inform the pollen diet of commercial bumblebees, as well as the selection of plant species used in agri-environmental schemes and conservation. So
wow, that was fascinating. That's a lot of stuff you did.
yeah, it was a lot of fun.
That's great. What was your favorite part of this summer's program?
I really enjoyed kind of the collaborative environment of my lab, being able to work with my mentor, with different lab mates. We kind of all helped each other on our projects. So like I couldn't have done this without my mentor, Julie, and everyone in my lab. And I helped them and they helped me, and it was a really good time.
That was great. And do you think this project will impact what you do in your career?
I think so. I definitely like the ecology sort of fieldwork vibe, and I definitely want to stick with that going forward.
All right, that's great. Well, thank you so much for sharing your research with us.
Yeah, thank you for listening.
Yeah, I hope you have a good rest of your summer before the school starts.
Thank you as well.
Okay, that's our last poster here in person. So I guess we'll throw it back. I think there's going to be like 5 or 6 virtual presentations of posters on Zoom, and they will start at 3 o'clock. So please join us back here at this URL at 3 p.m. Thank you.